Okay, so glad to be here today, and I'm going to talk about our ICTAS project, which is looking at coupled biophysical biochemical systems to remove predominantly nitrate from groundwater, and we're looking at nitrogen speciation and nutrient mass reductions with these systems. I'd like to point out the collaborators, so Mark Reiter and then Emily Lassiter, who's really the brains and the brawn behind this project. She's a graduate student working on it and has done some really interesting work, which we'll talk about. So just a quick outline of what we're going to talk about. First, we'll talk about some of the background and what this system is, the denitrifying bioreactor description, some design and applications, research objectives, methodology and testing, and then some results, both quantitative and qualitative results. So what is a denitrifying bioreactor? Basically, it's a system that we've designed and installed in agricultural fields that fosters denitrification in situ. So denitrification is a microbial stepwise conversion of reactive nitrogen in the form of nitrate to inert dinitrogen gas, 78% of the atmosphere. So basically converting bioavailable nitrogen to inert nitrogen. You need anaerobic conditions for this to occur. You need an organic carbon energy source to foster these reactions. And nitrate serves as the electron receptor in this pathway. So the motivation for this really comes down to the Chesapeake Bay total maximum daily load regulation, which is in effect right now. We are basically required to reduce nitrate, phosphate, and sediment inputs to the Chesapeake Bay by nearly 30%. So we really need to figure out ways that we can meet these standards, while obviously keeping agricultural productive. And the motivation was that coastal ecosystems and estuaries are heavily impacted by this reactive nitrogen or nitrate. And we think that there's really a high potential for removing nitrate from these ecosystems and agricultural systems. So basically in this diagram, we're focusing on the denitrification step, so conversion of nitrate stepwise to denitrification to nitrogen gas. So what is this denitrifying bioreactor system that we've developed? This is sort of a schematic of some of these systems we've developed and what they look like. So essentially what we're doing is in tile drain fields, that is fields that have subsurface drainage to remove excess water so you can get proper plant growth. We've installed these systems on the outlets of the tile drains, and we're able to control the water table both in the field, so we can, during the summer when there's usually moisture stress, we can back the water table up in the field to provide excess moisture for the plants and improve plant growth. But then during times of excess moisture, for instance, right now, we can fill this bioreactor with this high nitrate concentration groundwater and treat it effectively in situ. So when we talk about performance of these systems, it's generally well established. They've, over the past 10 or 15 years, there have been several studies that looked at sort of nitrate reduction efficiencies in the system. And they generally perform very well, up to 90% reductions in nitrate. The problem is that really these studies have only looked at inlet concentrations and outlet concentrations of nitrate. So what's flowing into these systems and what's leaving the systems. They don't really account for any conversions or alternative mechanisms that might be reducing nitrate to a less bioavailable form. And we're really focused on greenhouse gas emissions in these systems, so methane, nitrous oxide, nitric oxides, and trying to kind of quantify their impact in a larger system. They're pretty well established, especially in the Midwest. This is sort of the first system that's been installed in the Chesapeake Bay. And like I said, what we're trying to do is distinguish between other ENS transformations and like full denitrification, which is really what our ultimate goal is. So our objective is to maximize denitrification and minimize these other deleterious intermediaries like nitrous oxide, methyl mercury, or methane production. The first two are our greenhouse gases. And we're doing this through sort of substrate engineering. So can we engineer substrates for these systems to promote full denitrification and perhaps have activity on other compounds? So design and implementation, what do these things look like? So fundamentally, they're just big holes in the ground. And in this picture, what you're seeing here is the surface expression of one of these big tile drainage systems, draining a 30-acre field. So we excavate some big holes, essentially, line them so we can get kind of complete complete system. We don't have fluxes outside of the system. Fill them with these different carbon substrates. So in these cases, we're looking at two different substrates. One to the left is just wood chips, that's the carbon source. And the other one is wood chips plus a biochar, which is fundamentally an organic carbon similar to a activated carbon substrate, which we postulate have impacts on other contaminants, as well as perhaps promoting denitrification. And so this is our experimental setup. We've been conducting a series of both controlled experiments where we pump in a known concentration of nitrate-laden water, as well as natural events. So when we get a big rainstorm and we get the tile drainage system responding, we're out there to quantify reductions in that case. And so just to set up here really quick, we have the inlets, which is noted on the figure, and then outlets for each of these systems, the wood chip only and the wood chip plus biochar. This allows us to control the water table level in the reactor, maintain residence times, and look at sort of redox gradients across the system. Then we have these wells that you see in the middle of this one bioreactor here, and this is where we're doing passive gas and headspace diffusive gas sampling. We also have a system in the upper right-hand corner you see there. It's a SCAN system, and it's a real-time, high-resolution carbon-nitrogen analysis system. We can measure TOC, DOC, nitrate, chemical oxygen demand, water temperature, and water level, all in real time at high resolution. So it gives us a real nice image of the temporal dynamics across an event. So first, since this is probably the simplest part of it, I'm going to show something about the substrate testing. This is the biochar. So we speculated initially that it would promote denitrification through three mechanisms. First, soil water retention. It's essentially a dust that we've added to these systems, so it does increase the available water capacity or the water retention time in these systems. And this is really interesting. We also found that it adjusts the pH in the aqueous phase of that system, so it increases the pH of the system, which we've actually just kind of produced some initial results showing that can help push denitrification all the way to completion and actually reduce nitrous oxide production in these systems. And finally, this is ongoing work. We're looking at sort of enhanced microbial growth as another means of promoting denitrification. We've also looked, or we're just starting to look at some of the potential to remove other contaminants, for instance, phosphorus, pesticides, and certain pharmaceuticals in groundwater. Especially in this area, we have a pretty high burden of poultry antibiotics in some of the groundwater systems, so it'd be nice to be able to try to start to remediate them in situ. And what this figure is showing here, on the left axis we have the nitrate concentration, and on the right axis we have the orthophosphate concentration. In groundwater, to the left, in runoff, and then after it's exited these systems. So this is sort of qualitative evidence of the different reductions. We can't really tell what is occurring in this system at this point, but we can definitely see some pretty dramatic reductions in both nitrate and phosphate, up to 90% in some cases, but usually on average about 75% reductions, which is pretty significant. Here's some more kind of qualitative evidence for denitrification. Clearly, these systems on the left-hand graph here are pretty adept at removing nitrate from that waste stream. So this is a time series of an event just last month, beginning of this month, where we measured the nitrate concentration in the bioreactor as a function of time for the two different sort of systems, biochar and wood chips. And again, you can see the temporal dynamics of nitrate removal. We speculated it's denitrification, but at this point, or with this data, we can only tell that it is actually being either removed or converted to another form. And again, we consistently saw across events like these about a 75% reduction in the nitrate concentration in this system. These two graphs on the left are showing the redox potential and the dissolved oxygen. And really that's just sort of more evidence showing that the conditions in these reactor systems are conducive to denitrification. So the redox ranges shown here are what you'd find in the literature for denitrification to be occurring at sort of its optimum range, as well as as well as the dissolved oxygen concentrations. And you'll note that both of these tend to fall as the nitrate is reduced or converted to another form, which is pretty typical of what you'd see in laboratory studies in literature. So this is finally the final slide on the kind of qualitative evidence for denitrification. We've done quite a bit of isotope analysis looking at the N15, O18, N2O, and N2 ratios to try and get at, is denitrification actually occurring in these systems? So in this slide, what you're seeing here is that denitrification, or really the stepwise reduction of nitrate to N2, is discriminating against heavy N15. So we've observed this in our systems, and that the dissolved N2 has a negative enrichment ratio with the heavy N15, meaning that it's lighter compared to the atmospheric N2 concentration. So this basically means that the difference between the number of N15 atoms versus the atmospheric N2, the more dissolved N2 is present the relatively heavier it gets, reflecting that denitrification must eventually convert sort of that nitrogen from the heavier pool that's discriminated against once it's reached its sort of lower limit on denitrification. And indeed, so in some groundwater studies and lab studies, people have seen some similar relationships. This is from Wilson et al., which is sort of some of the seminal groundwater work looking at denitrification relationships. So again, the greater N2 concentration, if you looked in the literature, the greater N2 concentration might be expected to correspond to greater progression in denitrification reaction along that sort of multi-step reaction. But what we're actually seeing is that you can't really use these fractionation factors for the entire reaction. So the reaction is nitrate to nitrite to nitrous oxide to nitrite oxide to N2. And if you look sort of in the literature, what you see is that there's one fractionation factor for the reaction that's proposed to cover that entire reaction. And really, what we're seeing is that each of these different steps in denitrification can have a different sort of fractionation factor. And this is just a little bit more evidence of that here. So this is showing that denitrification also discriminates against the oxygen 18. And so this linear relationship between O18 enrichment and N15 enrichment is really widely observed during laboratory studies. This is sort of the first evidence we've seen in field-based studies of this type of relationship. There is some from the groundwater literature, but groundwater is a much more confined system. Our system is much more open to the ambient atmosphere, so we thought this was kind of interesting to see this same type of reaction in a less confined system.
Zach, we're going to need to be wrapping up in about another minute or two, please.
Okay, that's about what I have. So we've also been looking at the quantitative measures of denitrification through direct measurements. So we're measuring dissolved N2 and N2O with a headspace analysis where we collect a water sample with minimal atmospheric exposure, inject helium into the headspace and equilibrate the dissolved and aqueous phases, and then perform a GCMS analysis to look at the different constituents. And Emily actually developed a pretty novel GCMS method to detect first all the noble gases heavier than argon, as well as some of these other constituents we're interested in. So we're using argon as an inert tracer, and we can use that to distinguish between atmospheric and then biochemically produced dinitrogen based on the variation in that ratio. And then the N2O to N2 ratio, we're looking at that in response to these environmental parameters like dissolved oxygen, redox potential, temperature, etc. And then finally, the N2O emissions are estimated by comparing them to the IPCC indirect agricultural emission factors. So this is just sort of a screenshot of the GCMS analysis showing that we can, in fact, discriminate all these different sort of gas phases in sort of at levels lower than ambient atmospheric concentrations, which we thought was important. And this just shows the ion chromatograph scan of that same sort of analysis, showing where the H fall on the ion chromatogram. So again, direct quantification of denitrification occurring. And then finally, just the highlights from this study. We've got 2 papers which we're readying for submission, 2 extension publications as they come out of it as well, which are important. And that would end my presentation.